an explanation of the long COVID single system thesis, Part 1, the Endocannabinoid Command and Control System, a primer for clinicians. Endourage uses biotechnology to modulate the endocannabinoid system through safe and effective therapeutics for use as adjunctive therapy by healthcare providers. In a gold standard level of proof, Endourage conducted a randomized, placebo-controlled clinical trial of a phytocannabinoid-based therapeutic in long COVID. Participants experienced significantly reduced sleep disturbance and anxiety. Other assessed symptoms, including pain, fatigue, brain fog, dyspnea, social roles, and depression, also improved. No significant adverse effects were observed. This is the first evidence-based treatment shown to be effective for long COVID. Here's why. The endocannabinoid system, the ECS, is highly conserved in evolution. It includes the largest collection of neurotransmitters and receptors that are densely packed throughout our brains and bodies. This command and control system regulates and controls many of our most critical bodily functions, such as learning and memory, emotional processing, sleep, temperature control, pain control, inflammatory and immune responses, and eating. The ECS is at the center of renewed international research and drug development. The ECS has three core components: receptors, endocannabinoids, and enzymes. The cannabinoid receptor type 1, or the CB1, was discovered in 1988 by Alan Howlett and William Devane as the most plentiful neurotransmitter in the brain, which led to their collaboration with Dr. Meshulam in the discovery of anandamide, a naturally occurring endogenous cannabinoid in the human body in 1992. In the brain, the CB1 receptors are abundant in the cerebellum, basal ganglia, hippocampus, and dorsal primary afferent spinal cord regions, which is why cannabinoids influence functions such as memory processing, pain regulation, and motor control. In the brainstem, the concentration of CB1 is low, which may be why phytocannabinoid use is not associated with sudden death due to depressed respiration. CB1 receptors are also found in the spleen, white blood cells, endocrine glands, and parts of the reproductive, gastrointestinal, and urinary tract. The other predominant receptor, the cannabinoid receptor type 2, or CB2, is also reasonably abundant in the brain, but is much more evenly spread throughout the entirety of the nervous system than CB1 receptors. In 1993, the CB2 receptor was discovered in the heart, blood vessels, white blood cells, kidneys, bones, gut, tonsils, spleen, reproductive organs, and lymph cells. In the immune system, CB2 regulates cytokine release. The CB2 receptor regulates a variety of critical processes in the peripheral nervous system. CB2 receptor activation exerts significant control over pain and inflammation. Anandamide and 2-AG are the most widely studied endocannabinoids that bind the two specific receptors, CB1 and CB2. Anandamide also interacts with the vanilloid receptor. Enzymes are responsible for metabolizing endocannabinoids. There are two major enzymes responsible for this. One, fatty acid amide hydrolase, which metabolizes anandamide, and two, monoacylglycerol acid lipase, which typically metabolizes 2-AG. Terpenes are hydrocarbons, which are responsible for the scent and taste of all plants. Several terpenes have now been shown to act directly on the cannabinoid receptors themselves. Beta-caryophyllene was the first terpene to be identified as a cannabinoid receptor ligand. It works as a CB2 agonist in the human body. Citral has been shown to work via the CB2 receptor as well. So far, all of these terpenes have shown activity on the CB2 receptor. Presently, we have no data on a terpene which is a CB1 ligand. Flavonoids are a group of natural substances with variable phenolic structures found in fruits, vegetables, grains, bark, roots, stems, flowers, tea, and wine. These natural products are well known for their beneficial effect on health, and efforts are being made to isolate flavonoid compounds. Flavonoids are now considered as an indispensable component in a variety of nutraceutical, pharmaceutical, medicinal, and cosmetic applications. This is attributed to their antioxidative, anti-inflammatory, anti-mutagenic, and anti-carcinogenic properties, coupled with their capacity to modulate key cellular enzyme function. Research on flavonoids received an added impulse with the discovery of its favorable influence on cardiovascular mortality rate and prevention of CHD. More research is to come as it relates to the single-system Thesis for long COVID, but presently, the data supports the Thesis and the adjunctive therapeutic is safe, effective, and available.